Have you ever stopped to think about how the entire instruction manual to build you fits inside a space so small you literally can't even see it? Welcome everyone, let's jump right into this explainer. We're going to uncover the fascinating history of genetics and then explore the mind-blowing physical architecture of our DNA. We'll trace the journey from studying simple garden peas all the way to mapping out the incredibly complex, three-dimensional structures hidden deep inside our cells.
We've got a brilliant journey ahead in this explainer. We'll start with the mystery of heredity, discover the transforming principle, crack the genetic code, and then wrap up by looking at how both prokaryotes and eukaryotes package their genomes.
Section 1. The Mystery of Heredity. Let's look at how this whole field kicked off with a monk, a bunch of pea plants, and, honestly, a massive amount of patience.
Before we hit the historical timeline, let's quickly ground ourselves with two words we're going to be using a lot. First up, gene. It actually comes from the Greek word gen, meaning to create or give birth. It's the absolute basic unit of hereditary. Then we have the genome, a term coined back in 1920 by a German botanist named Hans Winkler. This is the complete master set of genetic information an organism needs to function. Think of a gene as a single sentence of instructions and the genome as the entire master blueprint.
You know what's really wild? How long Gregor Mendel's work just sat there. He presented his pea plant research in 1865, proposing that distinct factors were inherited from each parent, and then he passed away completely unrecognized. It wasn't until 1909 that Wilhelm Johansen finally called these factors genes. After that, things absolutely skyrocketed. By 1928, Frederick Griffith stumbles onto this crazy transforming principle that allowed harmless bacteria to suddenly become deadly to mice. And then, the bombshell. 1944, Avery, McLeod, and McCarty proved this mysterious transforming principle wasn't a protein at all, it was DNA.
Moving on to Section 2, Discovering the Transforming Principle. How did scientists actually prove that DNA and not protein was doing the heavy genetic lifting?
This brings us to the famous Hershey-Chase-Waring blender experiment. It's such a clever biological puzzle. Alfred Hershey and Martha Chase used radioactive isotopes to tag viruses. They essentially painted the viral DNA with radioactive phosphorus-32 and the protein coat with sulfur-35. They let these viruses attack some bacteria and then, literally using a kitchen blender and a centrifuge, spun them apart. The result? Only the glowing radioactive phosphorus made it inside the bacteria. The sulfur protein stayed outside. Boom! DNA was officially the inherited material. It was an incredibly elegant way to settle one of biology's biggest debates.
But look, science is all about correcting mistakes, right? Phoebus Levine was a brilliant guy. He figured out DNA had bases, sugars, and phosphates, but he totally missed the mark on the structure, thinking DNA was just a boring, repetitive polymer with equal amounts of adenine, thymine, cytosine, and guanine. Enter Erwin Chargaff. He crunches the numbers and proves that actually the amount of adenine always equals thymine and guanine always equals cytosine. The total purines exactly match the total pyrimidines. Without that one crucial math correction, Watson and Crick never would have figured out the double helix.
All right, Section 3, Cracking the Genetic Code. Let's decode how a simple four-letter alphabet translates in the insane complexity of life.
So, as scientists like Nirenberg and Khorana figured out in the 1960s, DNA is read in triplets called codons. Basic math time. 4 nucleotides read 3 at a time gives us 64 possible combinations. But we only use 20 amino acids to build proteins. So why 64? It's nature's built-in fail-safe. Multiple different codons can ask for the exact same amino acid. It's a built-in redundancy that ensures if there's a tiny typo in your DNA, it doesn't automatically cause a catastrophic protein failure. Pretty smart, right?
Now we're shifting gears from the history of time to the biology of space. We've got a serious geometrical mystery on our hands here. If you took the DNA from just one of your cells and stretched it end to end, it would be about 2 meters long. That's over 6 feet. Yet it fits into a nucleus so small you can't see it without a powerful microscope. How on earth is that physically possible?
Let's find out in Section 4, Prokaryotic Genome Packaging. We'll start with the ultimate minimalists of the cellular world, bacteria.
If our eukaryotic cells are sprawling, cluttered mansions, prokaryotes are ultra-efficient, tiny homes. Bacteria don't bother with a membrane-bound nucleus. They just pack their circular DNA into a dense little region called the nucleoid. They bundle their genes tightly together into groups called operons so they can be read all at once, and they carry almost zero junk DNA. Meanwhile, eukaryotes like us have multiple linear chromosomes, we wrap our DNA around histone proteins, and we hoard vast amounts of non-coding repetitive sequences.
Even in a simple E. coli bacterium, the packaging is wild. Its single chromosome is 4.6 megabases long, way bigger than the cell itself. So how does it fit? The cell physically twists the circular DNA up like a rubber band wound way too tight, creating these intense supercoils, and they organize it all into distinct spatial domains separated by protein barriers. This means if one loop gets relaxed or damaged, the rest of the loops stay tightly coiled and protected. It is a total masterclass in spatial efficiency, compacting it all 1,000-fold. Which brings us to Section 5, Eukaryotic Chromosome Architecture. Let's scale up to our own cells and see how this packaging gets infinitely more complex.
To picture this microscopic engineering, imagine a sewing kit. You've got long threads and little spools. That's exactly how our negatively charged DNA interacts with positively charged proteins called histones. Opposites attract, right? So the DNA naturally and tightly winds itself around a core of 8 histones, looping about 1.6 times around each one. This forms a structure called the nucleosome, which literally looks exactly like beads on a string.
And this is just step 1 of a massive hierarchical organization. Those beads on a string, they get coiled up into a thicker 30-nanometer fiber. Then, that fiber gets folded into loops averaging 300 nanometers. Those loops get compressed and folded even further into 250-nanometer fibers, which finally coil up tightly to form those dense mitotic chromosomes you probably remember from biology textbooks. It's like twisting a string, then twisting the twisted string, over and over again.
Let's put some numbers to this because it's absolutely mind-blowing. A single human chromosome 22, if stretched out, is about 1.5 centimeters long. But your cell crushes it down to just 2 microns. That is a 10,000-fold compaction ratio. 10,000! Without these specialized histone and non-histone proteins successively folding and coiling our DNA like a master origamist, complex life literally couldn't exist. The geometry just wouldn't work.
Now, inside the nucleus, it's not just a random alphabet soup of floating genetic material. It's a highly compartmentalized jungle, and every chromosome claims its own specific real estate. We call these chromosome territories. When a cell is just doing its normal daily business, chromosome 1 has its designated corner, chromosome 2 has its spot, and so on. They don't just float around aimlessly. This strict 3D geographic organization is absolutely crucial to keeping your genome stable and making sure the right genes are accessible at the right time.
If we zoom into those territories, the DNA is divided even further into what we call topologically associating domains, or TADs. Think of TADs as local neighborhoods on a chromosome. If a neighborhood's genes are turned off or repressed, the cell literally pushes that entire TAD to the outer edges of the nucleus, right up against the nuclear wall. But if a gene needs to be actively read, its TAD gets pulled into the busy, bustling center of the nucleus where all the transcription machinery is waiting. It's dynamic, structural gene regulation in real time.
So I want to leave you with this incredible paradox to chew on. The very packaging that protects our blueprint creates a massive, almost impossible hurdle for life to overcome every single second. Think about it. Your genes are buried under 10,000 folds of compaction, wrapped around histones and locked into specific territories. Yet somehow, fast as lightning, your cells effortlessly unzip the exact right sequence, read the code, and zip it back up without tangling the 2 meters of thread inside. It is an absolute architectural miracle happening inside you right this very second. Thanks for hanging out with me for this explainer. I hope you leave here with a brand new appreciation for the blueprint of life.